Good morning, good afternoon, good evening, depending on wherever you may be. Welcome all to our Facilitating Generic Drug Product Development through Product-Specific Guidances webinar. My name is Lieutenant Commander Nora Lim, and I am with the Small Business and Industry Assistance Program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communications. And I will be your host for today's session. If you haven't already, we encourage you to get connected with us. For your convenience, we've included QR codes to access SBIA on LinkedIn, Twitter, and YouTube, and via email updates. Scan the QR code with your camera on your cell phone. This will provide you with a link to SBIA on the platform for the QR code that you scanned. It's a great way to stay connected with SBIA social media platforms to learn about upcoming webinars, conferences, and regulatory information, and also to find recordings of past webinars and conferences. Now let's turn our attention to some webinar logistics. Please note, this event is 100% remote, in addition to being 100% virtual. Our speakers and panelists, not to mention our SBIA team and technical staff, are in separate locations. As such, there may be some internet issues, but please bear with us. And if in fact we do encounter those, we'll try to get them resolved as quickly as possible. Speaking of tech issues, here's our link for our one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. That's the same page that you used today for your online access to the event, so please bookmark the page. If you have any technical questions about this webinar, please check there first and you will most likely find the answer. We also invite you to submit your questions to us so that we have a vibrant and productive Q&A segment, which is so crucial to these events. This is a learning opportunity, and we encourage you to take full advantage of the expertise offered here. When you do submit your question, please identify that individual presenter by prefacing your question with the speaker's name, as shown here, @Joseph, for example. Please keep in mind that whilst we may not be able to get to your individual question, as some of these sessions can literally have over 100 questions, they are always very helpful as we plan for future events. The final survey is the mechanism by which you'll receive the certificate of attendance, which will support continuing education or CE credits for REPS, SOCRA, SQA, and ACRP. When an attendee completes the survey or evaluation, they can then download a certificate of attendance, which may be used in support of those credits, and the certificate must be downloaded immediately after completing the survey, otherwise the opportunity is lost. Even if you do not want or need a certificate, please take a few minutes to complete the evaluation, as your feedback will help guide the design of our future events. Note that the evaluation and certificate are only available for 2 weeks, which means that the deadline date is May 9th. And also note that our recordings will be posted a couple days after the webinar as well. Now let's take a moment to call your attention to some of our upcoming events. Listed here, we also invite you to visit our CEDAR Learn webpage, where you can find links to SBIA tutorials along with other educational resources, many of which offer CEs. And again, that's at www.fda.gov/CEDARLearn. It's now my pleasure to introduce today's presenters. First, we'll hear from Dr. Joseph Kotzabar, a Regulatory Health Project Manager within the Office of Research and Standards within the Office of Generic Drugs. We'll also hear from Dr. Qi Zheng, a lead pharmacologist within the Division of Therapeutics and Performance 2, as well as Dr. Heather Boyce, a lead pharmacokineticist in the Division of Therapeutic Performance 2. Other presentations will be from Dr. Karthika Natarajan, a staff fellow within the Division of Therapeutics Performance 1 within the Office of Research and Standards within the Office of Generic Drugs, as well as Dr. Jihong Shang and Leah Falade, a senior staff fellow within the division, and Leah Falade, a senior pharmacologist within the Office of Product Quality Assessment 2 within the Office of Pharmaceutical Quality within the Center for Drug Evaluation and Research. So Please join me in welcoming our first presenter and presentation.
Hello everyone, thank you for attending the SBIA webinar titled Facilitating Generic Drug Product Development Through Product-Specific Guidances. My name is Joe Kozibar, and I'm a Regulatory Health Project Manager within Office of Research and Standards, Office of Generic Drugs. I'll be giving the first talk titled Product-Specific Guidance Program, Updates and Overview of Available Resources. In today's presentation, I'll first be covering the Product-Specific Guidance, otherwise known as PSG, background. We'll discuss an overview of the FDA PSG program, the GDUFA 3 commitments, the PSG prioritization and revisions. We'll then talk through public requests and public comments on PSGs. And lastly, we'll end covering the available online PSG resources. So we'll start with the basics first. What is a PSG, or product-specific guidance? So very similar to how the FDA publishes general guidances for industry on a variety of topics, FDA also publishes product-specific guidances that reflect the FDA's current thinking and expectations on how to develop a generic drug product that is therapeutically equivalent to a specific reference-listed drug, or RLD. These PSGs are going to contain product-specific recommendations that will help identify the methods for developing generic drugs and generating the evidence that is recommended to support ANDA approval. And these PSGs will include both key science and research output. Overall, the PSGs are a very unique aspect to the generic drug development program. But when we think about showing a generic product is bioequivalent to the reference-listed drug, you can't solely rely on the product-specific guidances. You should also take into account the rules and regulations, such as in 21 CFR 320, as well as the FDA's published general guidances for industry that were previously mentioned on the slide before. Starting in 2007, the FDA has been continually publishing PSGs, which provide clear and direct recommendations to ANDA applicants. You can see over time, since the beginning of GDUFA in 2013, PSG development has steadily been increasing over time, with fluctuations from year to year on published new and revised PSGs. As of today, there are 2,187 PSGs available on the FDA PSG webpage. 40% of these PSGs are for complex products. When we refer to complex products, we are specifically talking about the classification of approved new drugs as complex products for generic drug development purposes, indicated in MAP 524010, with the link on this slide here. One of the biggest driving factors with when PSGs will be published is the GDUFA, or the Generic Drug User Fee Amendments 3 commitment on PSG development. For non-complex NCE new drug applications, or NDAs, approved on or after October 1st, 2022, that's the start of GDUFA 3, a PSG will be issued for 90% of such NDAs within 2 years of the date of approval. This should seem familiar because this is no change from GDUFA 2 that started in 2017. So what this means is that for any new chemical entity, which are typically type 1 new drug application approvals, a PSG will be issued for 90% of these NDAs that are non-complex within 2 years of the approved date. What's different with GDUFA 3 are for complex products. For these complex products, again, approved in NDAs on or after the start of GDUFA 3 in October 1st, 2022, A PSG will be issued for 50% of such NDAs within 2 years and 75% of NDAs within 3 years. So what does this mean? This essentially says that for all NDAs that are complex products and approved after GDUFA 3, a PSG will be issued for half of them within 2 years, an additional 25% within 3 years, and then a remaining 25% of complex NDAs will not get a GDUFA 3 goal date. These are PSGs that are generally going to need research before a bioequivalence approach can be recommended. FDA will also continue to develop PSGs for complex products approved prior to the start of GDUFA 3, for which no PSG has yet been published. When you think about PSG prioritization and development, there are several factors that lead to initiating a PSG, as well as several factors that can prioritize how these PSGs are grouped into batches for publication. So some of the initiating events that will generate a new PSG or a revision PSG are recently approved NDAs or supplemental NDAs, FDA analysis of products without PSGs and if any gaps exist, pre-ANDA meetings and public requests, which we will discuss on upcoming slides, comments submitted to the PSG docket, controlled correspondences, and citizens' petitions. How PSGs are prioritized into batches are through a variety of ways, such as GDUFA 3 commitments, stakeholder interest in ANDA submission, drug availability and accessibility, public requests from generic drug industry and other stakeholders, as well as public health priorities. Lastly, there's data that are used in order to develop and publish these PSGs. Some of this data is taken from pharmacokinetic and pharmacodynamic modeling studies, previous bioequivalence studies, NDA review and labeling, pharmacovigilance, and last but not least, GDUFA-funded research outcomes. One of the questions you may have is how or why product-specific guidances get revised. So there are several factors that will lead to a need to revise a PSG. Some of these factors can be seen on this slide to the left. They include changes to the reference listed drug, such as a labeling update, a new supplement approval or a new strength approval, newly identified safety concerns, consistency with revisions to general guidances, responses received in BE comments, citizens' petitions, new bioequivalence approaches developed from research, and new knowledge from ANDA assessments. Overall, when a PSG is revised, there are going to be categories that the revision falls under. These categories are seen here on the slide to the right. They are critical, in vivo major, in vitro major, minor, and editorial. For critical revisions, the PSGs being revised include additional bioequivalence studies or evidence recommended to support the FDA approval that reflects safety and efficacy of the drug product. The critical revisions are the most important and will have potential to impact on all ANDAs, even including approved applications. In vivo major and in vitro major are PSG revisions that will include additional bioequivalence studies or evidence recommended to support bioequivalence and FDA approval. Last but not least, minor and editorial revisions will clarify studies or add additional language to the PSGs to help streamline generic drug development. When should you expect PSGs to be published? Overall, new and revised PSGs are published on a quarterly basis. We post PSGs frequently to a very specific schedule in February, May, August, and November. However, a PSG may still be published as a standalone guidance or a standalone batch outside of these quarterly scheduled postings. Some reasons for a standalone guidance or a standalone batch include coordinating with a citizen's petition response, meeting a GDUFA goal date, efficiency in developing PSGs for products in the same class, such as the topical batch of PSGs that were published at the end of 2022, and a level 2 revision. The FDA will issue a notice in the Federal Register for every batch and standalone posting, except for a level 2 revision. So you may be asking, why not a level 2? So what is a level 2 revision? So when we think about how PSGs are typically revised, these are going to be a level 1 revision. In fact, all PSG revisions until the end of 2023 were level 1 updates. This is where the PSG will be updated to describe changes in FDA's earlier interpretation or policy that are more of minor nature. A level 2 PSG revision is a typographical error found in PSGs only. There's going to be no change in the bioequivalence recommendation or agency thinking on the required studies needed to show bioequivalence. 3 level 2 PSG revisions were published for the first time at the end of 2023 in November. One PSG that was revised in November of 2023 through this level 2 revision process was ruxolitinib phosphate topical cream. Here you can see the typo that was identified in this PSG, and it was promptly corrected through the level 2 process. There was no change to the overall bioequivalence recommendation or agency thinking. This was simply a change in the API listed due to a typo. After PSGs are published, the public is welcomed to comment on these guidances. The FDA issues a Federal Register notice announcing the availability of new and revised PSGs via docket number FDA 2007D0369. The notice will identify a comment period for the recommendations. Comments can be submitted both electronically to the docket or by mail, and users can request additional assistance with a help desk ticket. The FDA will consider comments on draft PSGs while revising the guidances. If the PSG is not available, the public can request one via the CDER Direct NextGen collaboration portal. The FDA will review public requests received and take an appropriate action. You can see, since the beginning of GDUFA 2 in 2017, public requests have drastically declined. You may be wondering what's the difference between a controlled correspondence or a public request if both are submitted through the CDER Direct NextGen collaboration portal. Controlled correspondences received are triaged as public requests for reference-listed drugs that the submitter is requesting or proposing general bioequivalence recommendations, and the RLD has no published PSG. If the RLD is for a complex drug product, the submitter may also send a pre-ANDA meeting request. A public request response from the agency will typically say the request for information related to the product-specific recommendation for generic drug development was sent to ORS for further processing. ORS will take your request into consideration when publishing the product-specific guidance. However, you will not receive additional direct notification of guidance development from the agency. A great resource to use to identify upcoming PSGs, either new or revised, is this generic drug product development list, otherwise known as the forecast list. This is a list that's going to describe the FDA's plans for all upcoming new and revised PSGs of generic drug products in the next 12 months. What's new with GDUFA 3, this forecast list now includes both complex and non-complex products. Prior to GDUFA 3, this list was solely limited to complex products only. This list also enhances the transparency in PSG development for revisions and plans for generic drug products. What's new with GDUFA 3? Updates include a specific projected PSG publication date in a month-year format. This will clearly indicate the estimated month the PSG will be published. Prior to GDUFA 3, no such recommendation was given. The list also ensures consistency in FDA recommendations decisions following previous iterations of the PSG and establishes principles for PSG revision that reflects the most accurate, sensitive, and reproducible approaches. New in GDUFA 3 is we have redefined revision classifications that were previously mentioned on previous slides. Lastly, the forecast list is updated every quarter when a new PSG batch is posted. Here you can give the forecast list a quick look. There's going to be some key information to see. The forecast list is grouped by new and revised PSGs on separate lists, with complex and non-complex products grouped together within the new and revised lists. Here you can see some of the added information, such as the product complexity and the planned publication date. I want to note here, some of the products will list within the next 12 months or beyond 12 months. These are going to be products that are pending regulatory or scientific discussions, and the exact month and year of posting is pending. These will be updated as we get closer to the PSG planned publication date when it's ready for public posting. So now we'll shift gears a little bit and talk about the available online PSG resources. One of the most important available resources is the PSG website, otherwise known as the PSG database. This can be found through the FDA's website, searching under all drug databases. As noted earlier, there are 2,187 posted PSGs currently listed on the database. There is an alphabetized search function that users can search by the first letter of the API name. Importantly, at the bottom of the website, there is a section for newly added or revised PSGs from the most previously recent batch. Currently, you can see PSGs posted on February 15th, 2024. These are the PSGs that will be displayed here at the bottom of the website. By clicking on the dropdown for newly added guidances since February 15th, 2024, users can see all of the PSGs published in that batch. Here you can see the list of new guidances that were published, sorted by pages of 10. If users want to view the PSG page itself, they can simply click on the hyperlink linked to the API naming to pull up the PSG page. Users should see a standardized header template with the active ingredient name, dosage form, route, the strengths listed, and recommended studies. I will note here that PSGs that are not updated to this format are still accurate, and they can still be followed for generic drug development purposes. There are various sections of the PSG page itself. Next couple slides will discuss navigating the product-specific guidance. The first section of the PSG page will be the regulatory preface language. I'll note that this language has slightly changed over time, so older PSGs may not have this language listed as such. However, the bioequivalence recommendations in those older PSGs are not affected and can still be used accordingly. The next section of the PSG page is the core bioequivalence recommendations. For this PSG page, the recommended studies were 2 in vivo bioequivalent studies with pharmacokinetic endpoints, recommending both a fasting and fed study. After the bioequivalence study recommendations, there are several other sections often noted in PSGs. One of these sections is the dissolution recommendation. Here you can see the dissolution test method and sampling time section of this PSG. You'll often find a link to the dissolution database in the FDA's website. There's a talk coming up in this webinar that will be discussing the dissolution recommendations further. Another section often in PSGs is the additional information section. Here, users will often find drug device recommendations or additional product development recommendations discussing formulation updates. Again, we'll have a talk coming up in this webinar that specifically talks about the drug device recommendations often found in PSGs. Lastly, there's an administrative tracking section at the bottom of the guidances. This information is for internal purposes only and really should not be used. It's important to note that the unique agency identifier numbers should not be used as the RLD product the PSG is referencing. It's important to use the FDA's website, the database, in order to see which RLDs are being covered by a specific product-specific guidance. Here is an example of what was just discussed. Again, it's important to use the reference listed drug or reference standard number that's specifically put on the PSG database. These link directly to the Orange Book listing of the drug product. Through the Orange Book, users can find the active ingredient name, the proprietary name, the dosage form and route of administration, the strengths listed, if the product is considered a RLD or RS, the application number, approval date, and applicant's full name. It's also important to note we have the recommended date listed on the PSG database. There was a change to how these dates are listed recently. Prior to GDUFA 3, FDA only had listed the month and year the PSG was published. Now, specific days are listed that the PSGs were displayed. Using the RLD or RS information on the FDA's PSG webpage is important to help identify the products related to a specific PSG. A PSG may cover multiple RLDs, and ANDA applicants must identify the RLD appropriately. The listed RLD RS numbers are not a substitute for the Orange Book. Information is current when the PSG is posted, but the RS can change over time. Applicants should always use the Orange Book to confirm the correct basis of ANDA submission and the current reference standard product. Last but not least, we'll discuss GDUFA 3 meetings. New with GDUFA 3 is a PSG TCON that can be submitted if a change in vivo study has occurred and applicants have already began their in vivo study. PSG meetings, either pre-submission or post-submission, can also be requested following the PSG TCON if additional discussion is still needed. This allows a forum to discuss the scientific rationale for an approach other than the approach that's recommended in the current PSG. Pre-submission PSG meetings can be requested if the ANDA has not been submitted. Post-submission PSG meetings can be requested if the ANDA has been submitted. Alternatively, a controlled correspondence is a good way for applicants to follow up with the FDA on remaining issues following the PSG TCON. Other pre-ANDA and ANDA scientific meetings are also available as alternatives to PSG meetings. Here is a summary of the PSG TCONs and meetings that was posted with the GDUFA 3. Lastly, here are all of the resources used to compile these slides. These other resources can be used to learn further regarding the PSG program. That wraps up the first talk on an overview of the PSG program and available resources. Please stick around for further talks that clarify specific sections of the PSG page and goes into further detail on the scientific content. Thank you.
Good afternoon, everyone. Today, I'm going to discuss the tailored PSG recommendations for immediate release drug products and share our insights how the tailored PSGs optimize our approach to ensure therapeutic equivalence. The objectives of today's presentation. We aim to deepen our understanding of the specific considerations for PSGs and discuss the need for tailored PSGs through case studies, and also gain insights and explore strategies on how to apply these recommendations effectively. Let's look at the general guidance that frame our discussion. The FDA's 2021 draft guidance on B studies with PK endpoints. This guidance is foundational for anyone planning to submit an ANDA. The general guidance is applicable to IR, MR, or other dosage forms when systemic exposure measures are pivotal for establishing BE. The general guidance provides a framework foundation. However, each product design is specific and requires specific guidances. PSG reflects FDA's specific expectation for generics and highlights the FDA's approach to B studies for specific products. As you may know, for oral IR drug products, 88% of PSGs recommend in vivo B studies with PK endpoints. What specific considerations are included in PSG recommendation? Each PSG development considers a variety of factors from the label information, biopharmaceutics and PK properties, product design and development, and formulation and manufacturing characteristics. Those considerations are very important for B study design, subject selection, and for determining the appropriate and effective B studies. Certain drug products require specific regulatory approaches to ensure they're safe and effective when produced by different manufacturers. These include complex drug substances or complex drug products. Some specific BE challenges or issues also require alternative BE study designs and approaches. For example, BCS waivers for highly soluble drug substance, BE studies in patients, or PSGs based on approved suitability petitions. Some of those specific topics will be covered in the next presentations. I will focus on the general aspects of those tailored PSG recommendations. Starting with the selection of the appropriate strains and dose used in B studies. Under the general PK/B guidance, the highest available strains can be typically used as a single unit for BE testing. This standard applies across all IR dosage forms, tablets, capsules, or granules for suspension. However, as you will see, it might require some adjustments. For instance, concentrated suspension, or when we're dealing with bioanalytical sensitivity issues. In such cases, we can administer multiple units to ensure accuracy for BE measurement, but the dose must stay within safe or labeled dose ranges. For instance, oral suspension, the dose can be followed the recommended dose as stated in the labeling, taking into considerations that the dose administered is safe to healthy subjects. There are also situations where using the highest strength itself is impractical, perhaps due to safety issues or other limitations. We have alternative approaches to resolve those issues. One approach is to conduct a study with patients who are already taking the drug at this strength. Alternatively, we may consider using a lower strength in healthy subjects as appropriate if PK is linear. Beyond just adjusting strengths, there are also considerations when to test multiple strengths of the same product. Certain drugs exhibit nonlinear PK or there are significant difference in formulation among the strengths. In these cases, it's prudent to use a bracketing approach in BE studies, testing both highest and the lowest strengths to demonstrate BE. Let's begin with our first case study on gabapentin tablets. The PSG recommends BE studies for both lowest 300 mg strength and highest 900 mg strength, and a waiver for the middle strength, complemented by comparative dissolution testing and alcohol dose dumping. So the rationale behind these comprehensive evaluations stems from the unique formulation design and PK properties. Gabapentin exhibits nonlinear PK, and formulation is designed to retain the drug in the gastrointestinal tract longer than typical standard formulation. This slow release mechanism and the lack of compositional proportionality across the strengths necessitate robust and varied testing to ensure all strengths are therapeutically equivalent. Moving to our second case study, levothyroxine capsules. The PSG specified the use of multiple units of the highest strength 0.2 mg. The adequate BE on the 0.2 mg at a single 0.6 mg dose support the waiver of other 11 lower strengths that are not tested in the in vivo BE studies. For levothyroxine, accurate measurement of T4 concentrations is very critical to ensure detectable levels above baseline T4. 0.6 mg is recommended. This approach not only addresses the unique properties of the product and PK properties, but also aligns with the maximum recommended dose for both safety and effectiveness. Now let's discuss the selection of analytes in BE studies. General PK/BE guidance typically recommends demonstrating BE based on the parent drug. However, the selection of which analytes for BE purpose depends on several factors. These include the sensitivity to identify formulation differences, clinical importance, and whether it's a prodrug, and available analytical technologies. Each factor can influence the accuracy and relevance of the BE study. It may become necessary in certain cases to determine BE by measuring not just the parent drug, but metabolites. This is particularly true with prodrugs that are immediately metabolized after administration, and therefore it is not reliably measurable in plasma. In some cases, the parent drug undergoes significant pre-systemic metabolism, impacting the safety or efficacy of the drug products. Those require a more detailed PK profile to ensure a comprehensive assessment of BE. One specific example is enzalutamide tablets. The PSG specify measuring both the N-conjugated and its glucuronide metabolite. Ezetimibe undergoes significant pre-systemic metabolism into ezetimibe glucuride. Because of extensive hepatic recycling, the exposure of ezetimibe is less representative to evaluate its absorption. In this case, the total ezetimibe should be measured to ensure that BE studies reflect true therapeutic equivalence. Let's look at a broader landscape of BE studies. Depending on the product characteristics, different types of studies may be required to fully capture BE. The types of BE studies include single-dose fasting or fast studies, and even more complex setups like comparative clinical endpoint BE studies. They also include in vitro BE studies and bioequivalence options. Although standard BE study types cover most situations, there are scenarios where additional specialized BE studies are required. For formulation designs such as solid dispersions, lipid-based formulations, a single standard BE study might not suffice. Other dosage forms such as chewable tablets, complex drug products such as locally acting, might interact differently within the body, which need tailored BE studies. Gastroirritative or those with acid modifiers also need specific studies to accurately assess their BE. Now let's examine a specific example where multiple BE study types are important, using palbociclib tablets as our case study. The PSG recommends conducting BE studies using 3 distinct conditions: fasting, fat, and with an acid-reducing agent. These different testing conditions are recommended to fully assess the product performance across various physiological environments. The need for conducting BE studies with and without acid-reducing agents arises from its pH-dependent solubility. Originally, the capsule formulation required the use of ARAs with food to manage dissolution variations. The new tablet formulations, which include a pH modifier agent, shows improved dissolution and different behaviors under these conditions. The discrepancy between the capsule and tablet formulations makes it necessary to test both with and without ARAs to ensure the features of the tablet formulation can be reproduced in the generic products, and the generic products can handle varying gastric pH levels as effectively as comparator products. Successfully implementing such specific PSG recommendations requires a strategic approach. To ensure these complex and tailored PSGs are effectively put into practice, we are working on refining our existing BE guidance to incorporate the latest scientific findings and address new product design challenges. We also continuously update our risk assessment frameworks to capture, better predict, and mitigate potential risks associated with new generics. Moreover, ongoing communication with the pharmaceutical industry is critical. We use various mechanisms to improve and promote communications, such as regular meetings, workshops, conferences, guidance documents, to ensure these tailored recommendations are clearly understood and correctly implemented. These collective efforts support the safe and effective of generic drugs, facilitate their timely approval. To conclude, let's engage with a challenge question to test your understanding. Which statement about PSGs is not true? The answer is D. D does not reflect our discussion in this presentation. That concludes my presentation. Thank you very much for your attention. I will be back for the Q&A session.
Good afternoon. My name is Heather Boyce. I'm a lead pharmacokineticist in the Office of Research and Standards, and today I'll discuss development of generic drug products under Suitability Petition. Our objectives today include discussing changes permissible via Suitability Petition, understanding how the GDUFA III Reauthorization Act impacts Suitability Petition submissions and approvals, summarize the effect of GDUFA III on suitability petitions submitted since FY 2024, and discuss the structure of product-specific guidance for products approved via suitability petition. Permissible changes included that of a different active ingredient in a combination product, a different route of administration, although to date a petition seeking such a request has not been submitted, a different dosage form, and a different strength. GDUFA III Reauthorization provided goal dates for suitability petitions submitted between FY 2024 and FY 2027. I've broken down what those goal dates are. Starting in 2024, FDA commits to reviewing 50% of submissions in 6 months. Note there is a cap of 50 petitions eligible for this goal date. By 2027, FDA commits to reviewing 90% of submissions in 6 months with a cap of 90 petitions. As of March 1st, FDA has received a total of 77 petitions. Note, October 1st, 2023 starts the FY 2024 clock. And as you can see, 54 petitions were submitted at that time. A breakdown of the content of petitions submitted include 32 petitions seeking dosage form switches and 52 petitions seeking strength changes. If you're observant, you'll note that those two numbers add up to more than 77. Several of these petitions request both dosage form change switches and strength changes in the same petition. Of note, 15 petitions request a change in tablet to orally disintegrating tablet, which is the most frequent dosage form switch, and 18 petitions request a strength change for intravenous injection. And typically, these changes are requested for more patient-convenient dosing or to eliminate pharmaceutical waste. For FY 2024 petitions, FDA has issued a response to 28 petitions. And note that this meets our commitment in the GDUFA III reauthorization, which is to respond to 50% of petitions submitted in FY 2024. Of the 28 responses, 13 have resulted in granted petitions. Of the 13 suitability petitions granted, 1 is related to a tablet to an ODT switch, and 12 are related to strength changes. Okay, so let's dive into the PSG development on strengths approved via suitability. So I have an interesting case example, a cefixamine oral suspension. This product has 3 strengths that were actually approved via 3 different approval pathways. As you can see, this guidance is written to include bioequivalence recommendations for all 3 strengths. So let's dive into what happened. So for this case study, back in 1989, cefixamine oral suspension was developed in a 100 milligram per 5 milliliter strength as a B1 application. It was originally listed as the RLD, but it is currently discontinued. A generic version of this product was developed by Lupin in 2004 under a 505J application. In 2005, FDA approved a suitability petition requesting a strength change for this product from 100 milligrams per 5 milliliter to 200 milligram per 5 milliliter. The bioequivalence approach selected for this product approval, for the strength approval, was to compare the ANDA product, which was identified as the reference standard at the time because the RLD product was discontinued, and the dose tested was 200 milligrams. So this was 10 milliliters of the 100 milligram for a 5 milliliter suspension tested against 5 milliliters of the 200 milligram per 5 milliliter proposed test suspension. And this resulted in approval of the 200 milligram per 5 milliliter. In 2013, a 500 milligram per 5 milliliter strength of this suspension was approved as a 505(b)(2) application by Lupin. And for this approach, the ANDA for the 200 milligram per 5 milliliter strength was selected as the listed drug. And the dose tested in the study was 200 milligrams. So that equates to 5 milliliters of the listed drug against 2 milliliters of the proposed new strength suspension. So FDA considers all three of these strengths as part of the same product line, even though they were approved under 3 different regulatory pathways. We assigned the 500 milligram per 5 milliliter strength as the RLD and RS for sulfexamine oral suspension. A waiver of in vivo testing is allowed for the lower strengths if certain conditions are met. Okay, so now we'll talk about development of dosage form changes via suitability petition. An example is a tablet to an ODT and pharmaceutical equivalent considerations. So when developing an orally disintegrating tablet, things to consider include taste masking, tablet friability and packaging, disintegration of the ODT, and intended label claims for petition ANDA that differ from the tablet. And why this is important, because this could result in selection of specific conditions for bioequivalence studies. And particularly, this can include whether or not you administer the tablet with or without water. For a tablet to orally disintegrating tablet change request, there may be special bioequivalence considerations to make. For instance, on the table on the left-hand side, you'll see what we typically recommend for an ODT product where the RLD label states the following. If you administer the ODT with or without water, we recommend applicants conduct the bioequivalence study without water. If it's to administer the ODT with water only, we state with water only should be used for the bioequivalence study, etc. On the right-hand side, you'll see a unique consideration there. If a petition ANDA labeling intends to state that the ODT should be administered with or without water, an additional study might be needed where the study is conducted both with and without water to demonstrate that the ODT product can work and be bioavailable under both conditions. And this may be unique only for the first petition ANDA to be approved. Subsequent ANDA submissions for that ODT product approved under suitability may follow the guidelines on the left-hand side once that first product is approved. So the future of PSG and suitability petitions. So what we would like to do is the PSG program will proactively support recommendations for approved suitability petitions. Traditionally, the FDA, as you saw in the sulfexamine example, has incorporated recommendations for additional strengths into the PSG only after a petition ANDA has been approved. Moving forward, FDA will attempt to proactively incorporate our recommendations for petition strengths into our PSG prior to a petition ANDA being approved. And I have an example of this, Primidone, a PSG that was recently published. In the future, you may see strengths and dosage forms in the PSG recommendation that may not appear in the Orange Book. And this will be because there is an approved suitability petition. So as you can see, an example, the primidone PSG, we've indicated which strengths as part of this product line have been approved via suitability petition. In this particular case, the 25 milligram and 100 milligram strengths are part of the approved suitability petition, but a product has not been developed for those strengths. However, our guidance is written to provide recommendations for how to develop those strengths should an applicant choose to do so. For our challenge questions, a permissible suitability petition request does not include which: a different route of administration, a different dosage form, a different strength, or a different sought form of a drug. Answer is a different sought form of a drug is not a permissible change allowed under suitability petition. For challenge question 2, by 2027, FDA commits to reviewing what percentage of suitability petitions in 6 months? 30%, 60%, 90%, or 100%? There is 90%. In summary, our GDUFA 3 commitment goals in FY24 related to suitability petitions have been met. So we met that initial goal date. We've already reviewed more than 50% of submissions received in FY24. Industry has successfully leveraged the revamped suitability pathway as evident by the high number of submissions received since FY 2024. FDA is proactively providing PSG recommendations for petition strengths. Dosage form changes may have unique product characteristics and special considerations to establish bioequivalence that should be considered. And as you saw in my slides, I gave an example of a tablet to ODT switch, and depending on the intended labeling claims of that ODT product, you may have to have special considerations for those studies. Additional strengths are likely eligible for a waiver of in vivo testing, although special considerations may need to be made. Thank you for your time today, and I'm happy to take any questions.
Good afternoon, everyone. Thank you for joining us today. My name is Karthika Natarajan, and I'm a reviewer with the device evaluation team in the Division of Therapeutic Performance 1 in the Office of Research and Standards within the Office of Generic Drugs at the US FDA. My talk for today will focus on the recommendations that are included specifically for device and user interface assessment in drug-device combination product PSGs or product-specific guidances. Let's start by first understanding what are drug-device combination products. Before we do that, we need to first understand what are combination products. A combination product is defined in 21 CFR 3.2 and is a product that is a combination of any of the following: a drug and a device, a biological product and a device, a drug and a biological product, or a combination of all three, a drug, a device, and a biological product. A drug-device combination product, as the name indicates, comprises of a drug constituent part and a device constituent part. However, a drug-device combination product can have either a drug or a device primary mode of action, depending on whether the drug or the device contributes most to the intended therapeutic effect or action of the combination product. Among the various centers within FDA, CDER regulates drug-device combination products with a drug primary mode of action, and that will be the focus of my talk today. For the remainder of the presentation, a DDCP or a drug-device combination product will refer to drug-device combination products with the drug primary mode of action only. A drug-device combination product is said to have a drug primary mode of action if it meets the definition of drug contained in Section 201G1 of the Food, Drug and Cosmetic Act and does not have a biological product or a device mode of action. A drug-device combination product with drug primary mode of action can be a single entity, co-packaged, or cross-labeled drug-device combination product. In a single entity combination product, the drug and the device constituent parts are physically, chemically, or otherwise combined. Example is a drug prefilled in a syringe. In a co-packaged combination product, the drug and the device constituent parts are packaged together. An example would be a drug prefilled syringe that is co-packaged with a needle. In a cross-labeled combination product, the drug and the device constituent parts are provided separately, but they are specifically labeled for use together. Product-specific guidances for all drug-device combination products, including the three types that we mentioned earlier, should include device and user interface related language irrespective of the complex or the non-complex nature of its device constituent part or parts. The device constituent part of the drug-device combination product is considered complex if the device design impacts drug delivery to the site of action or absorption. Example is a metered dose inhaler, and or the user interface requires additional training of the patient by a healthcare provider before the patient can use the product. Example, a device tracks the dose with an electronic system. This slide provides you some examples of complex device constituent parts. Besides drug-device combination products with complex device constituent parts, there are also drug-device combination products in which the device constituent part is simple, such that the drug delivery to the site of action or absorption is not impacted by the device design but is actually controlled by the user. Examples are dosing cups that are used to measure the volume of an oral liquid formulation, dosing cards that allow the user to measure appropriate dose of an ointment, pre-filled syringes with needles where the delivery is controlled by the user and not by the device itself, intravenous bags where the user controls the rate of the infusion, either using a clamp or an infusion pump, and eyedroppers where the user controls the number of the drops that are delivered to the eye. Now that we understand what a drug-device combination product is, let us now focus on the device and user interface language that is included in the product-specific guidances for these drug-device combination products. Before we delve into the actual device and user interface language, let's discuss some important dates regarding FDA, generic drug regulation, and drug-device combination products. In 1984, the Drug Price Competition and Patent Term Restoration Act, or the Hatch-Waxman Amendments, were enacted that increased the availability of less costly generic drugs to the public. It was 15 years later, in 1999, that the first guidance, a draft nasal guidance, was published that included device-related language to assist firms to develop their generic nasal product. This guidance was later revised in 2003. In 2012, there were three major events. GDUFA user fee program was established, OGD became a CEDAR superoffice, and the first product-specific guidance for an inhaler that included device-related language was published. In 2019, a separate device evaluation team to support pre-ANDA review of device and user interface was established within the Office of Research and Standards in OGD. And by 2021, the device evaluation team initiated PSG Device Language Project. The purpose of this project was to include device and user interface language to all drug-device combination product PSGs. Since then, the device evaluation team has been increasingly integrated into the PSG development process, and a collaborative review process with the other team within ORS has been established. The device evaluation team reviews and adds or revises the device and user interface language for the following PSGs: new drug-device combination product PSGs, draft or final drug-device combination product PSGs that are published but do not contain device or user interface language, draft or final drug-device combination PSGs that are published with device or user interface language but require revisions. For all new PSGs or PSG revisions, the device team evaluates if the PSG requires device-related language addition or revision. As a first step, the device team determines if the RRD for the PSG is a drug-device combination product or not. And if it is identified as a DDCP, then what are the device constituent parts of the DDCP? If either the DDCP status or the device constituent part is unclear to the device evaluation team, the device evaluation team sends an inquiry to the CDER product jurisdiction officer for input. Once the DDCP status and the device constituent parts are confirmed, the device team reviews the device constituent part of the RLD to develop the device language. Here is an example of a 2-page PSG that shows the location of the device and user interface subsections in relation to the other PSG sections. The PSG example included here is for a fulvestrant drug-device combination product that is available as an injection for intramuscular administration by a healthcare provider. Fulvestrant is an estrogen receptor antagonist used in the treatment of breast cancer. As you can see, the device and user interface language subsections are provided to the end of the PSG under the section Additional Information. We will discuss these subsections in the upcoming slide. The first subsection is the device subsection that includes a statement about how the RLD is presented and identifies the device constituent parts. Next, the aspects of the RLD device constituent part that a generic firm needs to examine when they design or choose their test device is specified. The second subsection is the user interface subsection. It includes standard language that was developed with OGD policy in fall of 2021 and is included in all current drug-device combination product PSGs. This section is intended to inform the industry that a complete comparative analysis between the generic product they are planning to develop and the RLD is expected. Now let's look closely at the language included for how the RLD is presented and what are the device constituent parts. For this particular PSG for fulvestrant, based on the current prescribing information, instructions for use, and the new drug application data, the device team identified that the RLD consists of a kit with 2 drug pre-filled syringes and 2 injection needles with needle guards. The 2 pre-filled syringes and the needle with needle guards were identified as the device constituent parts. Once the RLD device constituent parts are identified, the device team then identifies the features of the device constituent part that a user interfaces with. This includes general features such as the size and shape of the syringe, the needle and its needle guard, as well as its external critical design attributes and external operating principles. If the device does not have any unique features, the language provided in the PSG will be a standard language and will be as follows. Next, the device team will assess if there are any unique features of the device constituent part. The pre-filled syringes and the injection needle with needle guard were identified as the device constituent parts in the fulvestrant PSG. The pre-filled syringes for this product are used only once and then they are discarded. Each syringe delivers a fixed dose, 250 mg of the drug in 5 ml intramuscularly, and the drug comes pre-filled in the syringe from the manufacturer. Hence, the single-use, fixed-dose, pre-fill format are identified as unique features of the pre-filled syringes for this particular product, and they are included in the device language section. For the needle, the RLD's needle gauge and length are identified as unique features, as a change in the needle gauge and length might be perceived by the patient differently if the generic needle is different from the RLD product. The needle guard system is also identified as a unique feature, as the absence of a needle guard system in the generic might increase the risk of needle sticks and the overall risk profile of the generic product when it is substituted for the RLD. Let's now look at some additional examples of device language included in product-specific guidances. This slide will include example of device language included in PSGs for single product presentation of the RLD. First product is a drug administered by the ophthalmic route, and the second product is a drug administered via the vaginal route. The specific presentation of the RLD and its device constituent parts are first identified in the device language. The devices included here are simple devices with no unique features, and the device language in these 2 PSGs recommend that a generic applicant examine only the general features of the RLD device that we discussed earlier when developing their generic product. This slide will include some additional examples of PSGs with device language. The first example is a transdermal product. The RLD is a transdermal delivery system and it is a drug-device combination product. For this particular product, the size and shape were not identified as rate-limiting factors for drug delivery and hence are not included in the general features section of the device language. Next is an oral product. In this case, the oral dosage syringes are the device constituent parts. They are very simple devices and it is sufficient to examine the general features like size and shape and the volume markings of the syringes. There are no other specific external critical design attributes or external operating principles that needs to be examined for the RLD, hence they are not included in the general device features section of the device language. In this slide, we'll see some examples of device language with unique device features. It includes a complex pre-filled syringe system, a periodontal product with unique cannula features, and a simple rectal product with a unique lubricated rectal applicated tip feature. These are product-specific unique features that a generic applicant needs to examine when developing their generic product, in addition to the general features of the RLD. This slide includes device language examples for products with higher complexity, a nasal product and an implant, and shows the increased number of unique features of the RLD with increasing complexity of the device constituent parts. This slide includes an example of the device language included in the PSG for an RLD with multiple device presentations. The PSG device language identifies the unique features for each presentation that the generic applicant needs to examine and consider while developing their generic product. Do note that there might be separate PSGs for each presentation of the RLD for other products. This slide provides you some additional resources. In summary, all PSGs for CDER-led drug-device combination products should include device-related language. ORS is working to add device-related language to older PSGs for drug-device combination products. The device section identifies the device constituent parts of the combination product, while the user interface section directs the user to the guidance with detailed information to conduct a user interface assessment for their proposed generic referencing a drug-device combination product RLD. I'll be more than happy to take any questions during the Q&A session. As a closing thought, engage with FDA early and often to request feedback on your proposed generic drug-device combination product, device constituent part, and user interface through controlled correspondences or product development meetings. This concludes my presentation for today. Thank you for listening, and I will hand it over to the next presenter now.
Good afternoon. I'm Jiyoung Son, Senior Staff Fellow, Division of Therapeutic Performance 2, Office of Research and Standards, OGD CDER. I will be providing a brief overview as to what factors are generally considered and how they are assessed for the selection of study population in bioequivalence study with a PK endpoint. This is my disclaimer. In my talk, I will focus on major factors considered for the selection of the study population. My presentation will also introduce some decision-making framework on study population selection that OGD is currently practicing when developing or revising our product-specific guidance, PSG. This is the sentence in the section for study population selection described in the guidance for PKB studies for ANDA submission. Healthy subjects are generally recommended as a study population for PKB studies, unless the drug have any serious safety risk to such subject. All PSGs that the agency published carries a recommendation on study population when a PKB study one option for the approval of ANDA. This recommendation intends to warrant safety and acceptable tolerability in study subject for BE studies from clinical and ethical perspectives. When developed each PSG, clinical safety assessments are conducted to support a recommendation on the selection of study population. The safety assessment process focuses on addressing all potential safety concerns under the circumstance of BE study in healthy subject using an intended BE dose. These are major considerations for the selection of study population when developing PSG. Non-clinical toxicology profile and associated risk are basic review information. Drug-specific and drug class-specific safety profile is a key review component. Safety data from healthy subject is also important information for the assessment. As the last step, all safety concerns are assessed from the level of the drug exposure in healthy subject following administration of an unintended BE study dose. Generally, study population selection has been determined on a case-by-case basis because each drug has its own unique safety issue and different level of data availability. The clinical assessment team for PSG in OGD has been developing review framework to assess each consideration factor for standardized review process. The first consideration factor is a non-clinical toxicology component. Cytotoxic agents are generally recommended to conduct BE study in patients. However, we have PSGs of some cytotoxic drugs which recommend healthy subject. Amphotericin B, nalburtimox, and venetoclax are examples. The decision of each drug was made through rigorous assessment from toxicology and clinical perspectives. When generic drug developers conduct a BE study of a cytotoxic agent, it requires bio-IND application without regard to study population to follow the requirement described in CFR. Genotoxicity and carcinogenicity are also key non-clinical safety components. The first two bullets on this slide present the representative toxicology tests that are reviewed primarily to address genotoxicity and carcinogenicity for the assessment. For each toxicity, the safety margin is estimated in relation to human recommended dose and unintended dose in BE study for the assessment. Reproductive toxicity from animal study also served as a key consideration. If a drug has a significant potential for reproductive toxicity, such as embryo-fetal development toxicity and or infertility potential identified in animal study, we could recommend restriction on study subject or including additional comment on contraception requirement during and after the BE study in PSG. When assessing reproductive toxicity, we look at the following four elements. The first element is embryo-fetal development toxicity. This toxicity is assessed using safety margins such as no observed adverse effect level, NOAEL, in relation to human exposure level. Fetal death cases in animal studies are taken into account as significant findings. The second element is the required contraception period. The contraception period for participants in BE studies can be estimated based on the available PK and the toxicity information. If an estimated contraception period for the recommendation is long enough, this option might not be a practical measure to prevent a potential pregnancy in healthy subject for BE study. In current practice, in a period of 1 month or longer is regarded to produce concern about preventing an unexpected pregnancy. Genotoxicity and infertility are also considered. When it comes to infertility, reversibility of the impact on reproductive organ and their function from animal studies is a key assessment point for the decision. We also collect information as to whether NDA program included subject of reproductive potential in healthy subject study. Finally, we make a recommendation related to reproductive toxicity by considering the totality of the evidence of all these elements. I'd like to present two examples to introduce the overall assessment practice of reproductive toxicity for PSG development. The first example is belumosity, which is a raw-associated cold kinase ROCK inhibitor and approved for the treatment of chronic graft-versus-host disease. The current draft PSG recommends excluding female of reproductive potential. That decision is supported by the following rationale. First, based on pharmacological mechanism of action of belumosity as a ROCK inhibitor, it would produce a harmful effect on embryo-fetal development. As the second reason, embryo-fetal development toxicities, including fetal loss cases, were reported in animals at the exposure level lower than that at the recommended human dose. Lastly, the phase 1 studies during NDA program also excluded female of reproductive potential. Thus, it was concluded that healthy males and healthy females not of reproductive potential are appropriate BE study population for belumosity. Second example is cabozantinib. This drug is a tyrosine kinase inhibitor and approved for the treatment of several advanced cancers. In animal studies, cabozantinib was found to cause a significant damage on male reproductive organs. This damage appeared to be possibly irreversible. From the toxicology assessment, it was concluded that there was no sufficient safety margin at single dose in relation to male reproductive organ dysfunction. Thus, we recommend excluding male of reproductive potential for B study of cabozantinib. Let me move to the next consideration factor. For study population selection, the safety profile of drug in human subject is the most essential review item. Clinical assessment mainly focuses on assessing if safety concern identified from human study still exists in healthy subject, even at single dose for B studies. These are the most common safety issues that lead to restrict the use of a healthy subject. Hepatotoxicity, including hepatic enzyme abnormality, heart rhythm abnormality, especially QT interval prolongation potential, hypersensitivity risk, and blood dyscrasia, which can be associated with infection or hemorrhage risk. When assessing each safety issue, two key questions should be addressed. Whether and how likely does a single dose administration of the highest strength cause clinical relevant event in healthy subject? Can any alternative option be considered? By addressing these questions, we first can decide on the appropriate selection of study population, patient or healthy subject. When we recommend healthy subject, we still include caveats such as study dose selection and/or additional safety comment in PSG as a precautionary measure. Generally, the following information and assessment can be used as a resource to support the study population selection to address safety profile in human subject. Major safety issue identified in the patient from NDA program is key information. All safety data from healthy subjects are also thoroughly reviewed. This data mainly come from NDA program. Data from NDA program and the public domain is also reviewed if they are available. Pharmacological plausibility is considered to predict possible occurrence in healthy subject. The assessment can include the relevant safety finding of the other drug, in the same class as part of the evidence. When available, previous regulatory discussion and the pharmacovigilance database are also utilized. From the assessment using all available clinical safety information from patient and healthy subject, we choose one or more of the following options as recommendations. As a first option, we can recommend the patient for PKB study rather than healthy subject due to seriousness of safety issue. There are many examples. However, in this table, I included the only one represented drug, pazopanib. The decision for study population selection of the pazopanib was made mainly based on the report of the severe hepatotoxicity event in the bioequivalence study conducted in healthy subject. As the second option, we can recommend the use of lower dose rather than the highest strength due to the safety reason, even though healthy subjects are feasible for B study. These three drugs, aripiprazole, bandetanib, bavacamten, are the representative examples which recommend the B study using a low dose in healthy subject. Each drug has its specific major safety concern, summarized in the table when the highest or higher doses are used in healthy subject. Thus, we recommend a lower dose or lowest strength of the available strengths for the therapy studies. As additional options, we can also include a specific comment for the exclusion criteria related to safety issues. These two comments are commonly included for PSG as an additional safety comment. Let me conclude my presentation. PSGs describe the agency's current thinking on the most appropriate options, including study population and those to ensure safety in subject for PKB studies. The current practice for the risk assessment for the selection of B study population considered non-clinical toxicology, safety data from patient and healthy subject, and plausibility assessment for each safety risk. The decision framework for B study population has been being developed based on up-to-date information and the outcome from the regulatory research and communications with generic drug developers. I'd like to thank everyone who contributed so much to establish the clinical review framework for the development of PSGs, especially clinical safety and human subject research team members, division and office management ORS, all collaborated within OGD. Thank you all for listening. I will address questions you have in the Q&A session.
Thank you, Dr. Sean. Hello, my name is Dr. Leah Filade. I'm a senior pharmacologist in the Division of Product Quality Assessment 12 in the Office of Pharmaceutical Quality Assessment 2. Today I'll be giving the last talk today in the webinar on the FDA dissolution methods and navigating the FDA dissolution database. The views expressed in this presentation are those of the speaker and do not necessarily represent the views and policies of the FDA. So I just want to go over a brief outline of my talk this afternoon. First, I'll be discussing the dissolution testing for quality control purposes. Then I will go through the FDA dissolution database, including on how to navigate the database. And then I'll give a brief summary. So let's start with the definition of dissolution. Dissolution is the kinetic process of a solid dissolving into solution. It's a prerequisite to drug absorption and in vivo performance for almost all solid oral dosage drug products. For the purpose of my talk, this testing is used for quality control to ensure the drug product quality over the shelf life and the batch-to-batch consistency is met. Next, I'll talk about dissolution method development for quality control purposes. The dissolution method is product specific, meaning that the dissolution method should be appropriate for your proposed generic product. The dissolution method should be sensitive to possible changes in drug product quality over the shelf life of the drug. We request that the dissolution medium be physiologically relevant. When submitting your application to the agency, at least 12 dosage units of each strength should be tested. For collecting the dissolution profile, the sample should be collected until drug release is complete, which means at least 85% is released, or a plateau is reached, meaning there are no increases over 3 time points. The dissolution data should have low variability, meaning the percent CV should be less than 20% at early time points and less than 10% at all other time points. The method should be validated and reproducible. Validation parameters should include, but are not limited to, specificity, linearity, accuracy, precision, and reproducibility. Immediate release solid oral drug products containing drug substances with high solubility are exempt from method development requirements, and standardized dissolution methods can be used. These methods are found in our Guidance for Industry, Dissolution Testing and Acceptance Criteria for Immediate Release Solid Oral Dosage Forms from drug products containing high solubility drug substances. These two methods outlined use either USP apparatus 1 basket at 100 RPM or USP apparatus 2 paddle at 50 RPM and 500 milliliters or 0.1 normal HCl. These media should contain no surfactant and the temperature should be 37 degrees Celsius. The acceptance criterion recommended is Q equals 80% in 30 minutes for these products. Next, I'll go through the FDA dissolution database. The FDA dissolution database is publicly available. To date, there are 1,518 dissolution methods that are published. The methods that are available are for several types of drug products, including tablets, capsules, transdermal systems, injectables, and oral suspensions, just to name a few. These methods should be used as a starting point for method development of your proposed generic product. The final method selected should be optimized and justified for use for quality control dissolution. Next, I will go through navigating the FDA dissolution database. Here you can see the main page, and first I will go through the highlighted links that I've circled here. The first link takes you to a printable version of every method in the database that is alphabetized by drug name. These methods can be exported to a CSV or Excel file. There's also a filter function available here. The database contains the drug name, dosage form, USP apparatus, the rotation speed, the medium, volume, the recommended sampling times, and the date that this entry was updated. These sampling times are only recommendations and will vary based on the dissolution profile of your proposed generic product. The next link is our FDA methods database disclaimer. I will not read it in its entirety, but in summary it states that the database reflects the methods used for quality control in approved applications that have been found acceptable by the FDA, USP, and FDA guidances. They are not requirements, and the FDA will consider alternate methods if supported justification is submitted with your application. We revise this database on an ongoing basis, and the Division of Biopharmaceutics welcomes any comments or suggestions. So next I'll go through an example. In this example, I can type in ibuprofen followed by search. So these are the results. In this case, there are 19 entries for drug products containing ibuprofen. This also includes any combination products in the search results. In this example, the tablet formulation already has a USP method, and the applicant is requested to refer to the USP for this dissolution method. This database contains dissolution methods. However, the acceptance criterion should be proposed in the A&D submission, and the adequacy is determined during the assessment of the A&D. As I mentioned before, the dissolution database is updated frequently. The dissolution database is updated anytime a PSG batch has posted. Applicants may also request a dissolution method to be added to the database by submitting requests through the external correspondence operation system. If the QC dissolution method is changed for a reference product, the NDA may update the dissolution method using a prior approval supplement. The database will be updated to reflect the revised dissolution method for the RLD. In September of 2023, a significant revision to the database was made. Methods using water as a medium were revised. The reason that we are no longer recommending water as a dissolution medium is because water is not physiologically relevant and water lacks buffering capacity. Therefore, these drug products, it's recommended that the applicants develop a dissolution method for their proposed generic drug product. So in summary, the published dissolution method should be used as a starting point for method development. And it's a reminder that the dissolution method that an applicant wishes to use for quality control purposes is drug product specific. Water is not recommended as a dissolution medium because it lacks buffering capacity and is not physiologically relevant. The adequacy of the method and acceptance criterion or criteria will be determined during the assessment of the ANDA. Here's a list of the resources I've referenced in my talk: the Dissolution Methods Database, the Dissolution Testing and Acceptance Criteria for Immediate Release Solid Oral Dosage Form Drug Products Containing High Solubility Drug Substances Guidance, and also our tablet scoring guidance. If the proposed generic product is scored, then split tablet data should be submitted and tested, and this guidance will provide recommendations for that. I would like to acknowledge Dr. Bhagwan Reddy, the Division of Biopharmaceutics Director, Dr. Okpa Erderi, the Biopharmaceutics Branch Chief, and the members of the Dissolution Database Working Group. We work hard to keep the Dissolution Database current and make these updates I've discussed. Thank you for your attention. If there are any questions, I'll be joining my other FDA colleagues at 3:20 p.m. for speaker Q&A.
Hello, everyone. Thank you for attending the SBIA webinar titled Facilitating Generic Drug Product Development Through Product-Specific Guidances. Now we're going to shift gears and move into the live portion of the webinar. So upcoming, we have a full panel discussion with a variety of topics that we'll cover. On the panel, we have David Coppersmith, a regulatory counsel within Division of Policy Development within OGD-P, Utpal Munshi, a division director within Division of Bioequivalence 1, Office of Bioequivalence, Markham Luke, a division director within DTP1, ORS OGD, Office of Generic Drugs, Myung Jin Kim, another division director within DTP2 in ORS OGD, Liang Zhao, a division director in the Division of Quantitative Methods and Modeling within ORS OGD, Lei Zhang, a deputy director within ORS OGD, and last but not least, Leah Falade, a senior pharmacologist within OPQ, Office of Pharmaceutical Quality. So we'll just jump right in to our first panel discussion topic. This topic is going to cover the BCS waiver approach in PSGs. So overall, the BCS-based bio waiver is an efficient alternative bioequivalence approach for immediate release IR oral solid dosage forms that facilitate generic drug development. However, there are fewer products that have BCS-based biowaiver options recommended in PSGs than the number of products that may be eligible and may be known as BCS Class 1 or Class 3 products from a variety of sources. So our first question to the panel is, what is FDA's approach in including the BCS biowaiver option in PSGs? And then how can applicants use the BCS biowaiver as an alternate bioequivalence approach that is not recommended in PSGs? For these questions, to kick off this topic, I'll invite Myung-Jin Kim, or MJ, from ORS OGD to kick off our discussion. Thank you.
Yeah, thanks, Joe. First of all, we are very delighted to be here to host this webinar on very important topic of PSGs. And I'm joined by very distinguished panel members, so we are excited to be here. And we are aware that more than 1,000 people have registered for this very important webinar. So with that said, in terms of the BCS, what I can tell you is that the FDA's current approach is there is a committee, FDA BCS committee, and when the applicant actually requests a BCS-based biowaiver, that request goes through the FDA BCS committee, and if the committee reaches a consensus that there is a classification that should be designated, then that gets included in our BCS database. So our FDA database has a good number of products that are designed or classified as BCS 1 or 3. And we do have our PSGs actually have that information as well for the BCS-based waiver. However, if there are some PSGs that are not updated, the plan is to have them updated at the time of our ICH-M 13 related.
Sorry, MJ. Sorry to interrupt. I think we may have lost your sound on your mic.
I'm sorry.
Thank you.
So this is the most okay. So I'm not sure where I got cut off. So just briefly repeat, the FDA has the BCS committee that we evaluate the applicant's request for the BCS classification, and we do have a good number of PSG, the products that are classified as BCS 1 or 3, and we revised we have updated PSGs with that information. And if there are lacking information in PSGs, we are trying to update those PSGs at the time of ICH-M 13 related PSG revision that we were discussing the topic 2. With that said, one very important the aspect is that if our PSGs are silent on BCS-based waiver option, that does not mean that it is not recommended. So that's very important to remember. So if the applicants, if you guys believe that your drug should be eligible for BCS-based waiver, then our request to you is that submit that request to our control correspondence approach in the pathway. And it's very important to pose a specific question, the right set of questions at the time of control correspondence submission. So for example, you should not pose a question as to is our product considered as a Q1 the same, Q2 similar, that type of question. But if you can be very specific as to can our product be eligible for BCS based waiver based on whatever the information that you have. So we really encourage you to submit that request in the control correspondence. And we do very much value the importance of BCS-based waiver option, especially for the oncology type of products where the BE studies may have to be conducted in patients, or they may be conducted with the clinical endpoints, which poses a lot of challenges. So we highly encourage you to consider this option by submitting your request in CSIS. So with that, I will turn it over to you. Thanks.
Yes, thank you, MJ. Did anyone else in the panel want to weigh in on this topic? Utpal.
Yeah, thank you, Joe. And I would just like to, I guess, maybe start off with a point that MJ made earlier, that just because a recommendation for BCS waiver options on the PSG doesn't mean that an applicant can't consider that pathway, so long as the criteria in the M9 are met, right, in terms of the characteristics of the API, the solubility characteristics, or permeability as well, if we're talking about a BCS class 1 based waiver, and then also your formulation in comparison to the reference product formulation, and then finally the dissolution. So, at the end of the day, the biowaiver, this type of biowaiver in particular, is based on very fundamental principles, and simply the fact that it's not mentioned, a PSG shouldn't preclude us or preclude you from attempting to pursue it. And I think the broader point too to consider is that, for example, that some of the information for BCS biowaivers is known, so you can use that to gain some confidence in terms of pursuing the pathway. For example, information on the RLD labeling, or just general knowledge of the chemistry of the API can give you some confidence to pursue that pathway. And then on top of that, as MJ said, there is the controlled correspondence pathway to get some further insight into the proposal. And the other, I think, more general point, of course, is whether we're talking about BCS-based biowaivers or anything else for that matter. Just because something is not recommended in the PSG doesn't mean that you can't or shouldn't pursue it. At the end of the day, if the science is satisfied and the regulatory requirements are satisfied, you still may end up with an approvable product. So just a few things to consider. Thank you.
To follow that, I will only have a very minor point to add, following what MJ and Utpal comments on the BCS biowaiver. So for some special cases, even the guidance haven't, as said, determined you can is qualified for BCS biowaiver. So if you have confidence that with your data, and also with quantitative approaches that integrate the data, for example, PBPK modeling, that can support some kind of biowaiver in special situations. In addition to controlled correspondence, you can also utilize a new mechanism like model integrated evidence industry meeting pilot to discuss the approach with us. That's another value you can utilize in terms of doing BCS-based biowaiver for products that's not currently identified with recommended with biowaiver option in the PSC. Thank you.
Yeah, thank you, Liang, very much for chiming in there. Okay, I think we can move to our second discussion topic. So this is a discussion topic that may be on a lot of people's minds. So this is going to revolve around the ICH M13A harmonized guidance. So what is the FDA's plan on PSGs that are not aligned with the M13A with regard to study recommendations after the M13A is finalized and ready for implementation? So to kick this off, I'll invite Lei to chime in first to kick off the discussion. Thank you.
Yeah, Thank you, Joe. As you may be aware in the presentation, but as Joe alluded to, it could be in many people's mind. And you may be aware that M13A, which is the ICH guidance on bioequivalence for immediate release solid oral dosage form, the draft was published in December 2022 and currently is under public consultation and a revision to reach to step 3, which is going to be the finalized guidance to be agreed or endorsed by ICH. So you probably see the draft format, and you may be aware, and is also alluded to this question, is that there's some things that impact FDA's current general guidance, as the PSGs, which are typically aligned with our general guidance, which is currently FDA recommend both fasting and Fed-B studies for almost all IR products, but in M13A, it is recommended a risk-based approach, which means a lot of products may not need to study this but be sufficient. So right now, the M13A is still in stage. Once final is anticipated to be later this year, and it's adopted by ICH, FDA as the ICH member will adopt this guidance. So we will as well we all the PSGs can be impacted by M13A, that will also be aligned with new thinking that's aligned with M13A once it's finalized. So this is in the plan at FDA. And we also may wonder how PSGs since we already have general guidance, but hopefully you guess is that PSG is really give us the opportunity to provide with tailored recommendations and all recommendations. We have the goals to develop those new PSG to develop that. Because of that, we need to be closely thinking, involving thinking, and update those PSG to align with our changes. And the FDA, people probably may aware that European medicines, the EMA, also have PSGs, and their PSG sometimes may not align.
Sorry to jump in here, but I think we're getting some connectivity issues with your mic. So let's turn it over to MJ really quick to continue on the discussion regarding ICH M13A, and maybe we can bring back Lei after her mic gets fixed. Yeah, thank you, MJ. Take it away.
Thank you, Joe. So Lei, I wasn't sure whether it was only my audio, but until Lei gets back, so as you know, the ICH M13 implementation is so it's gonna go forward once it's adopted and ready for the implementation. And given that ICH M13 is specific to the immediate release drug products, however, if you look at the ICH M13, there are some sections that's relevant for other dosage forms, such as the study design, these study populations, and all that. So what we have is the other parts of the ICH M13, such as the 13B or 13C, which relates to highly variable or additional strength and waiver, and an NTI type of drugs that are not covered in the ICH M13A. And our ANDA BE guidance has those topics, and so you should continue to utilize the information that's in the general BE guidance. But kind of going back to those PSG specifically related to the IR products, we are within the FDA, we have a group of people. We are going to be working on the PSG revisions so that our latest thinking is reflected in our PSGs when the ICH 13A is ready for the implementation. So we want to we don't want to create any confusions to the applicants. And so we are trying to kind of align the implementation of 13A as well as incorporating those guidance or recommendations in our PSGs for the IR products.
Yeah, thank you, MJ. A follow-up question I wanted to ask for the panel discussion is the FDA's current ANDA bioequivalence guidance is partially overlapping with the M13A with more content that will be harmonized in the future. What is FDA's plan on the revision of the ANDA bioequivalence guidance when M13A is finalized and ready for implementation in the future? I'll invite Dave to help address this question and kick off the discussion. Thanks, Dave.
Thanks, Joe. So as noted, many of the topics that are covered by the current ANDA bioequivalence guidance, which is the bioequivalent studies with pharmacokinetic endpoints for drugs submitted under an ANDA guidance that was published back in August of 2021, are also covered by the M13A guideline. And there are also some major differences between the two. In addition, there are some topics that are covered by the current ANDA bioequivalence guidance that are not discussed in the M13A guideline, such as modified release products and narrow therapeutic index drugs. So once M13A is finalized and adopted by FDA, it will reflect FDA's current thinking on this topic, and FDA will take the actions that the agency deems necessary to align the recommendations in the current ANDA bioequivalence guidance with the recommendations included in the final version of M13A to ensure that the recommendations are consistent and there's not any unnecessary overlap between the guidances. And as noted in the CEDAR guidance agenda, FDA is currently working on revising the current ANDA bioequivalence guidance. Thank you.
Yeah, thank you, Dave. Before we move on to our third topic, are there any other panel members that would like to chime in on ICH M13A? Okay, we will move on to our third topic. We do have a lot of good content still coming up. So this third topic is a very common question I feel like the PSG program receives. So this first question is, what should applicants do if there is no PSG on a specific product that they intend to develop? So to kick off this discussion on this topic question, I'll invite Markham to kick off our discussion. Thanks, Markham.
Thank you, Joe. I really appreciate the question. This is a common question, like you said. Applicants, if you see that there's no PSG there, you want to think carefully about how you're going to approach this, and you can request and attend a pre-ANDA meeting, providing information about your proposed pathway to getting there. You can look at similar products and think about how those were done and propose some of that and use that as part of your proposal, and have a informed discussion with us. So do the necessary research before requesting the pre-ANDA meeting with us. And thanks, Joe. If anyone else has any comments on that, please do chime in.
Yeah, Markham, a follow-up question, if you can continue on, is similar to if there's no PSG developed, what if there's no reference standard available for products that an applicant would like to submit a generic application for, an ANDA?
So this is also a good question. The reference standard is often listed in the Orange Book, but sometimes the Orange Book has a little catch-up time to do before a reference standard is declared if the RLD is taken off the market, for example, in which case you can request and ask about that through a control correspondence. You can also look at what other products are available in the context and submit to us in the pre-ANDA meeting whether something is okay to use as a reference standard. But ideally, I think you want to start off with looking at it as a possible query via control correspondence.
Yeah, following that topic, this Liang, if I can add one point, that for the in the scenario where the reference product or ROD reference standard is discontinued from the market, it's not available. In that case, you can resort to some historical data. For example, we had a presentation a couple of years ago in ACCP, American College of Clinical Pharmacology, regarding how to use modeling simulation to serve as the bridging tools to bridge the data of the discontinued reference against available product on the market. They may not be bioequivalent to each other, but with some quantitative calculation, modeling, simulation, analysis, we can derive a different B limit to bring a generic drug product on the market without the reference standard or ROD. So in that case, please feel free to have a proposal and discuss with us in the format of either Mark mentioned controlled correspondence. Now we also have something in place, the MIE industry meeting pilot. So that pilot program is poised to deal with complex issues for non-complex product, including product without a reference on the market.
Right. I just want to clarify that the pre-ANDA meetings is specific for complex products right now, and that this is focused on bringing complex products to marketplace. If there's non-complex products, there are other options available via the control correspondence, like I mentioned, and you can propose, for example, taking API in some format and using that as a reference in some cases. But these are things that you want to check with us via control correspondence before proceeding down that regulatory pathway.
Yeah, MIE industry meeting pilot is a program that being set up, to handle complex issues for non-complex products. If you have a resort to a modeling approach, that's also a good venue for you to pursue. Yeah, thank you.
And then we have that third bullet point. Sorry.
Oh, sorry. Go ahead, Mark. And you're about to say something, or?
Oh no, did you want to comment more on the second bullet point?
Yeah, I wanted to actually talk a little bit more about the third one. I think, I think we heard from Markham and Liang about, what we can do sort of in the pre-submission space if there are concerns around a reference standard not being available, or if an applicant decides to take an approach that's different than what's recommended in the PSG. Coming from the Office of Bioequivalence and a regulatory assessment perspective, I think one thing that our reviewers like to see is, particularly with these alternative approaches, is a description from the applicant about, for example, if you're doing multiple studies to support BE for some of these complex products that you described, for example, the relative importance of each study in the overall demonstration of BE. So in other words, please explain your approach to the review team and justify, why you're proposing the path that you are. I think the other thing that we sort of see in this space is more related to, I think, what can even affect more straightforward applications, which is data organization within the application. I think what makes that issue more critical here is that for these complex products, you have a lot of studies. So sometimes it's harder for our assessors to sort of track where all the information is located and what relates to what. So anything that applicants can do to help the review teams understand how all the data fits together, I think would be very helpful. And in that regard, for complex products, we do have the product pre-submission meetings as well. Finally, the other thing too, I think, would be that if, once you've gone through a pre-NDA meeting and you've received certain recommendations or advice from the agency, that if then when you decide to submit and you find that you want to deviate from that advice for whatever reason, please identify those deviations and explain why. That will also be very helpful for us. So I think, the bottom line at the end of the day is communication. And the more organized and the more you can justify your strategy to us, that will be very helpful for our review teams. Thank you.
Yeah, Paul, just thank you. That's very helpful. I think the understanding is that FDA put a lot of work into thinking through how the PSGs, what is the best BE approach to put into the PSGs. So if you're going to deviate from that, it's helpful for you to think through why we did it, why we're suggesting you do it that way, but also rationalize carefully why you're doing it in an alternative way. And if you're going to come to us with a control correspondence or a pre-end meeting, it's often helpful to have some data to go with that at our meeting so that we can have some discussion, some fact-based discussion on your alternative approach. Thanks, Joe. Back to you.
Joe, if you don't mind, I'm sorry, if you don't mind, I did raise my hand and I wasn't sure whether you saw me, but just a few more comments, and these points are really well taken and very extremely important to consider for the applicants. The one thing I can say from our end when it comes to PSGs, as one can imagine, with the lot of things going on, when we try to develop PSGs, we also consider the applicants or the public request for BE, the PSG development. And this topic was covered in Joe's presentation at the very beginning. So it's very helpful for us to know if there is a high level of interest from the applicant's perspective, if a PSG development is needed. So we look at the number of submissions or the requests that come in from the public when it comes to the PSG development. So we consider that very importantly as one of the key elements to consider. So if applicants are looking into the PSG development, if you are looking for that, please submit the public request for the PSG so that we can prioritize in terms of which PSG to develop first. With that, thank you.
No, that's a great point, MJ. And we do monitor those lists very carefully to see when they're coming in and what folks are proposing, want us to develop how we want to prioritize our PSGs for both complex and non-complex products.
Yeah, thank you everyone for chiming in on this discussion topic. Before we move on to our fourth and last topic, just making sure any other panel members want to comment on any of these questions posed this topic. Oh, I think, Lei, you may be trying to talk and you look like you're unmuted, but I'm not quite hearing you on my end. Well, we can move on to our fourth one, Lei. Keep working on that mic and maybe we can circle back and get some information from you at the end. So moving on to our fourth and final topic for now is going to regard PSG revisions and this PSG forecast list that was mentioned earlier today. So why does FDA sometimes forecast and plan revisions on the same PSG within a short period of time? So the forecast list is only indicating the next 12 months or 4 batches of PSGs. So what are the typical reasons for these revisions? So I would invite either Markham or MJ to kick off this discussion regarding the forecast list and these updated revisions. Thank you.
So I'm going to go off mute first while MJ's figure out how to unmute. The forecast list is helps FDA, helps industry look at what FDA is going to be putting out there, all right? And when we revise the forecast list, it's usually because we've discovered something, some concern from a regulatory aspect, or there may have been a shift in the makeup of the RLD product that requires us to think about how to do the revision a little different, or there's some other factor that comes in to play with regards to developing the PSG within that period of time that we originally sought to get the PSG out. MJ, did you want to chime in there?
Oh, yeah, thank you, Malcolm. So when it comes to the PSGs and the planning ways as well as the revision, it's really a complex process, because it doesn't, once we publish the PSG, it doesn't end there. And because some of the major factors can be considered from the new drug, what's happening in the new drug side, right? There may be a supplement NDAs that came in and it got approved. And so with the newer information being available, we utilize the information to update the PSGs. And so from our end, we think that when we believe that updating the PSGs so that the applicants have the most updated information for the drug development is more helpful than not revising the PSGs, and the applicants may be in the dark, and we don't want the applicants to be surprised with the information that could have been shared with the applicant. So there's that component, and yeah.
And also, most of the revisions that we make are minor revisions. I think that that's borne out, right, Joe? You're shaking your head, yes. And with that regard, there's not going to be. It's not expected that there's going to be a lot of impact on current submissions that are coming in with the minor revisions. But again, like MJ said, we're trying very carefully to get the information needed out there in as timely a manner. So we'll send something out, and then there may be additional information, but we're not going to let that hold the current PSG that's going out, hold that up. So we want to get that certain informational pieces, especially like a new PSG, out in as timely manner as possible. And then if there are additional minor revisions that will come down later, those will come out in a later period of time. And these are coming out on a quarterly basis overall. So we're trying to meet those quarterly deadlines.
Yeah, thank you, Markham. Thank you, MJ. And you're exactly right, Markham. So the forecast list is a great place to go and find the delineation between PSG revisions, the minor versus major. So I think that is a great resource for applicants to use to tell what the upcoming revisions to these PSGs are. I do see Lei is back with us. Lei, do you want to try your mic one more time to add in any to this, or possibly the previous discussion topic slide? I think it's still muted, unfortunately. Still not working. It's okay. So we'll move on. So the very last question we have really for this discussion topic is going to be another really common question I feel like the PSG program gets asked very commonly. So what should applicants do if a PSG revision occurs during a product development phase or after the ANDA is submitted? And a couple follow-up questions to this is how to determine that impact and how to obtain the guidance from the FDA. So I feel like this may have been briefly touched during that last explanation, but definitely going to open up this to the panelists if anyone wants to chime in here on what applicants should do if a PSG is revised during their ANDA submission.
Thanks, Joe. I can jump in first and then others can chime in. But as was mentioned, the upcoming PSGs for generic drug product development webpage, lists the revision category and also the nature of the anticipated revisions. And generally, FDA does not expect that minor revisions would impact an applicant's development program, while major revisions and critical revisions could have an impact. During the assessment cycle, the impact may be communicated to the applicant through a discipline review letter or a complete response letter. But one of the new things under GDUFA 3 is the PSG teleconference pathway. And PSG teleconferences are available both during the product development, the pre-submission phase, and after submission, the post-submission phase. And applicants can request a PSG teleconference if they have already commenced or completed an in vivo bioequivalent study to obtain FDA's feedback on the potential impact of the new or revised PSG on its development program. And during a PSG teleconference, FDA may state that there are no impacts on the applicant's development program, or that the recommendations in the PSG impact the applicant's development program and therefore recommend changes to the development program. Also, during a PSG teleconference, FDA may, if applicable, recommend a path for future communication with FDA, such as controlled correspondence, which we've talked about a lot today, but also pre-submission PSG meetings, post-submission PSG meetings, or other types of meetings to seek feedback from FDA to ensure that any proposed changes or additions to the applicant's development program would be an acceptable approach for demonstrating bioequivalence. Thank you.
Yes, thank you, Dave. Any other panel members want to chime in here on what should applicants do potentially if the PSG is revised for their submitted ANDA?
Yeah, Joe, some of that will depend on what the changes are and where they go and whether what's being impacted. So it often be a case by case play based on industry needs.
Okay, well, thank you to all the panelists. We can wrap up a couple minutes early and jump into the Q&A. I do think we have received a lot of questions in the chat, so thank you to all of the questions that would have been submitted during this webinar. Yeah, so we can jump back to Nora and we can move into the Q&A portion. Yep, big thanks to all the panelists. And yeah, we look forward to answering the questions. Thank you.
All right. Thank you again to all our presenters and panelists for their excellent presentations and insights. We'll now begin our question and answer panel. If you haven't had a chance to enter your question into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. All right, so now we have some questions coming in from the audience. We'll start with a question for Dr. Chi Zhang. Does the FDA take into consideration the availability of a new RLD or RS before designating it as the new RS in case of a new higher strength improved? If the new higher strength is not market available due to its recent approval, that an applicant is unable to obtain an adequate supply to perform bioequivalence studies, how can they?
So if there is a newly approved strains and the PSG has been already updated to include B recommendations for the newly approved strains reference standard, but this strain is in limited distribution due to its recent approval, you may consider an alternative study design or approach to establish BE. For instance, you could use a next higher market available strains of the RS for the BE studies, which may differ from the approach outlined in the current PSG. However, you must provide appropriate justifications with information and data for the proposed alternative approach. I want to note that the PSG can never cover all possible scenarios, so we highly recommend that you reach out to us and submit your proposed BE study design and approach through a control correspondence or pre-ANDA meetings. Thank you.
Thank you for that response. We'll have one more question for you, Dr. Zhang. Can I use a recently updated PSG of a similar therapeutic class PSG instead of the PSG of an old, not updated PSG of the same drug?
So for products containing the same drug substance, same API, such as different dosage forms, single or co-packaged or fixed dose combination products for the same drug, it is advisable to refer to the latest version of the PSG for the study design and approach, as the most current thinking and decisions or recommendations could apply to all products containing the same drug. However, as demonstrated in case study 4, prospective, even for the same drug with the same indication, PhD recommendations can differ due to the differences in formulation design. So if you encounter such discrepancies in terms of the recommendations and the requirements in the PSG among those products contain the same drug substance, we recommend that you reach out to us by control correspondence or pre-ANDA meetings for agency's clarification and feedback. Thank you.
Thank you so much for that response. We have another question for you as well. If the highest strength is not a therapeutic dose per label, the multiple units to be used as a single dose to make up therapeutic dose, is it still okay to use the single unit for BE studies, or is the agency's expectation to use multiple units to make up the dose, which is within the therapeutic dose range?
If the PSG does not specify the dose for a specific drug product, it is recommended to use a single unit of the highest strength as a single dose, reference our general B guidance. However, if multiple units are necessary, such as for the oral suspension dosage forms, you must provide information data to demonstrate that the selected dose is safe for administration to study subjects. Thank you.
All right, thank you so much for your responses again. Now we'll move on to some questions for Dr. Heather Boyce. First question, this is a post-approval question. So the RLD is discontinued if the reference standard RRS gets switched from one applicant to another close to the initiation of the BE study. When all the licenses and shipments are finalized based on the previous RS, will the FDA accept the BE data with the previous RS?
Yes. So I think this situation doesn't necessarily sound like a post-approval question, but more or less an applicant is getting ready to start their bioequivalence study and the RS has switched and what to do. I think, just in general, if, a product was labeled as the RS at one point, and you have the study set up to be using that specific RS, it should in general be acceptable to continue to use that RS product as long as you have a proper supply of that product to conduct your BE studies and your in vitro studies for your application and submission. I do think that it's always good to talk to the FDA about your specific case, and we have multiple pathways to do so. The control correspondence pathway, PSG TCOM pathway is now an option, and I think you can get immediate feedback in that specific case. Thank you.
Thank you for that response. Another question for you, Heather. In a PhD, say the 3 different strengths were included, though they were approved on different dates. Were these 3 strengths have the same formulation?
Yeah, that's a great question. So going back to the sulfexamine case example that I displayed, when we evaluate strength waivers for PSGs, we do look at the formulation components of the form of the RLD product across the strength to determine if a waiver would be acceptable. In this particular case, the formulations, all 3 strengths do have qualitative, all 3 of the strengths presented in that case study do have a qualitatively same formulation.
Thank you again for that response. One more question for you. Since the fiscal year 24 goals have been met, will the SP that were submitted in fiscal year 24 but not reviewed receive priority in the queue for the fiscal year 25 goals?
Yeah, that's an excellent question, and that's something that I didn't cover in my presentation given the time limit. But this is absolutely correct in that things that were not reviewed in FY24 will continue to receive priority going into FY25. So those petitions not reviewed in FY24 will be reviewed first in FY25 prior to any petitions submitted in FY25 will get reviewed. Thank you.
Right, thank you for that information. Now we have a question that came in for Dr. Ji-Hung Chun. First question, can healthy subjects be selected for a PKBE study while the PSG recommends patients as a study population?
First of all, we understand that the challenge when BE studies are conducted in patient. However, I'd like to restate that the recommendation on study population in PSG reflect the agency current standards and thinking from safety ethical perspectives. Thus, we recommend that generic drug developers follow the recommendation described in the PSG. And I also would like to note that the investigator and applicant are responsible for protecting the right and safety of all subjects when conducting this study in order to comply with the safety reporting requirement of CFR. And the acceptability of the submitted data will be a review issue. And I also remind that adequate justification in support of any alternative approach, including study population selection, should be provided as a part of the ANDA submission. Thank you.
Thank you so much for that response. Next question for you, Dr. Chun. How do you select the study population in terms of subject demography, sex, and age?
I would like to remind of the recommendation described in the PK ANDA guidance. And if a drug product is intended for both sexes and adult, including geriatric, the study should consider enrolling subject both sexes and a diverse age group, including geriatric subject. As I presented in my slide, there are some PSG which recommend restriction on the enrollment of subject in a single sex as a study population due to reproductive toxicity, even though they are used for the both sexes. And we also have the several PSG which recommend excluding geriatric subject due to safety issue. So for your selection study population, you can refer to the appropriate patient population in terms of sex and age and published PSG. Yet we found many studies which were conducted in one dominant demography. Again, we still increase that applicant attempt to enroll both sexes and subject in diverse age range to represent the approved patient population, unless you have a legitimate justification. Thank you.
All right, thank you so much for that response. Now we'll move on to some questions for Dr. Leah Falade. First question for you, Dr. Falade. How do you select the dissolution medium and other parameters for immediate or extended release drug products? And how often is the FDA dissolution database updated?
Yes, thank you for that question. That's a good question. So when you're doing your product development, you're pretty much selecting your conditions based on your results. So you can use the methods in the database as a starting point and optimize them based on your product. So you can optimize the medium, you can try different media, pHs, different surfactants, whichever gives you the most optimum results with the lowest variability and complete release. You can test different rotation speeds. And so those are just a few named selection parameters you can test. You can also select different volumes. So that is how you'll go about selecting and optimizing your method. And as far as the dissolution database updates, we update along with the PSG batch postings, which is quarterly, or it can also be a case-by-case basis. If a different PSG is updated outside the batch and we need to post the method for that, we will post for that. If someone submits a request for a dissolution database for a specific product, they can use the ECO pathway, and we will include that update as well in the database on a case-by-case basis. Thank you.
Thank you so much. Another question that came in for you, Dr. Folliday. It was mentioned that a PAS should be submitted in case the dissolution method was updated in connection with an NDA. Could you please elaborate more on this? Does this apply for ANDAs too?
Yes, that's also a good question. So yes, the prior approval supplement is for NDAs as well as ANDAs. So if an NDA is finalized, that means everything is finalized for that product. The analytical procedures, the finished product specifications, the end process specifications, everything is final. So any changes that are made on both sides, NDA or ANDA, you will have to submit to us those changes for us to assess them and approve them. So depending on the type of change and the level of change, it could be either submitted as a PAS, a prior approval supplement, or a changes being effective supplement. Thank you.
Thank you so much for that response as well. Now we'll address a question that came in for Joe. Joe, can a controlled correspondence be submitted for a clarification question on a drug product in absence of a PSG?
Yeah, thanks for the question. So overall, if a PSG does not exist and a controlled correspondence comes in asking for any clarification or a bioequivalence recommendation for that RLD, it's going to be automatically flagged as a public request and then triaged to ORS to track and use in PSG product prioritization. So these public requests are recorded and used when we think about PSGs moving into batches. But for this question, that would be the approach taken if the CC comes in for no PSG. Thank you.
Thank you, Joe. We'll do one more question for you. For a complex product, if a change is listed in the plan to revise PSGs where a study is being removed, is the industry expected to meet the current PSG or the plan to revise PSG, which will be updated in the future?
Yeah, thank you for this additional question. So this would potentially be a review issue. The applicants should proceed at risk. So they can come in with that original recommendation. But if it is a major, or I should say if it's a in vivo update to the studies, they're also welcomed to submit a PSG TCON. This is a new meeting approach that was added with GDUFA 3. Thank you.
Great. Thank you so much for that response. Now we'll move on to some questions for Dr. Karthika Nararanjan. First question for Karthika. Can the drug-device combination status for an RLD always be determined from the RLD's PSG?
Thank you for that question. The Office of Research and Standards within OGD includes the DDCP status and the device information in all PSGs that are being developed or revised for drug-device combination products since spring of 2021. And ORS continues to add this information to previously posted PSGs, but it's still a work in progress. So as of April 25th today, not all DDCPs PSGs have the device information. So if you're unsure whether an RRD is a drug-device combination product, you can always request a formal determination through the Office of Combination Products at FDA, or you can submit the question in a controlled correspondence to OGD. Hope that answers your question. Thank you.
All right, thank you so much. We'll do another question for you, Karthika. One second here. Okay. Should ANDA submissions for the DDCPs with non-complex device designs include comparative analysis?
A comparative analysis should be submitted as part of an ANDA submission for all drug-device combinations products, and it is irrespective of the device constituent part being either a complex or of non-complex nature. Thank you.
Thank you so much. We'll continue to look for other questions that came in here. We'll do another question that came in for Dr. Chi Zhang. If the highest single therapeutic dose is 100 milligrams, while the MDD is 200 milligrams, on what dose should the BCS solubility study be conducted on?
So in general, the BCA solubility study can be conducted using the highest single therapeutic dose per the labeling. Yeah, we can, those are the reference that you can refer to the M9 guidance. Thank you.
Great. Thank you so much for that response. We have a question that came in that could be either for Heather or Jihong. Why is FDA insisting an enrollment of female subjects in the BE studies of products, although the RLD label clearly mentioned that there is no significant gender effect on the PK of the drug?
So I can take that question. So as I mentioned earlier, we recommend that the sponsor choose a study population which represent patient population in their B study. That's the reason why we still recommend, sponsor try to put it effort enrolling, both sex. So depending on the approval indication, that is what the agency prefer and recommend. Yeah, that's the answer to your question.
Great. Thank you so much for your response. We also have some questions for some of our other panelists as well. We have a question specifically for Dave Coppersmith. If a sponsor of a specific RLD desires to submit to the FDA prior to the PSG issuance, information relevant to the bioequivalence recommendations that may be included in a PSG referencing that RLD, is a citizen petition the only manner available to make that submission? If not, could you please specify the alternative manners of submission?
Thank you for the question. So the citizen petition process is not the only way. So if you're interested in proposing bioequivalence recommendations for the agency's consideration, you can refer to the Good Guidance Practices Regulation, which is 21 CFR 10.115. And that regulation contains information on the ways and how interested parties can participate in the guidance development process. Thank you.
Great. Thank you so much for that response. We have a question now for Upal Munshi. The question is, even though FDA is not implementing the M13A, will FDA accept ANDAs with studies consistent with the ICH M13A bioequivalence for immediate release solid oral dosage forms at this time?
Yeah, thank you. That's a good question. Thank you. So, as I mentioned during the panel discussion, whether it's a PSG or guidance, those consist of recommendations. And so applicants are always welcome to propose alternative approaches so long as they justify them. So with respect to this specific question, even though our current guidance is not the M13A, that is not our current guidance of record, if an applicant feels that from a scientific perspective, they can justify their approach based on the principles of the M13A, then they should submit that justification, and then it would be a review issue as to whether that study would be found acceptable or not. Thank you.
Thank you so much for that response. Now we'll have one more question for Lei. If you disagree with a BE approach in a PSG, is it better to publicly comment on that PSG or use a different communication method such as controlled correspondence? And we may not be hearing audio from you, Lei, if you're speaking. Okay, if not, we can try to come back to you on that one.
Do you want me to try to take that question while Leigh's working it out?
Sure. Yes, go
Hi,
ahead.
this is Markham. So I would suggest in that case that they can submit their questions, their disagreement to the docket with information. If they're in the process of developing a ANDA and they think that the BE impacts their ANDA approach, then they can request a PSG meeting like we had discussed during our discussion points.
Great. Thank you so much for that response and jumping in as well. So we have another question now. This one will be addressed to Joe. What role does the innovator company play in the PSG development, if any?
Yeah, thank you for the question. It's a good question. So really, the innovator does not play a role in the PhD's development. However, the innovators can and sometimes do comment on the PhDs after they're published using the bioequivalence comment pathway through the regulations.gov website. So the FDA will take into account any comments posted to the PhDs once those PhDs are publicly available. Thank you.
Okay, thank you so much for that response. Now we'll move on to a question for Dr. Qi Zhang again. We are a generic drug manufacturer, and we are planning to develop a solid dosage IR tablet for a lower 100 milligram strength. The 100 milligram strength of the RLD is listed in the electronic Orange Book as the RLD and RS, but the PSG recommends 200 milligram, which is not listed in the Orange Book as the RLD or RS. Should we conduct a BE study when using 2 100 milligram tablets or 1 100 milligram tablet of the RS and test products?
If you only develop a 100 milligram strength, not the higher 200 milligram strength, you may conduct the B study using a 1 milligram tablet against 100 milligram strength of the RLD. However, in the future, if you decide to develop a 200 milligram strength, additional B study on the 200 milligram strength may be needed. Thank you.
Thank you so much for your response there. We'll continue with another question that came in for you. It was mentioned on slide 7 as studies of strength for study strength and dose, the highest strength applicable for all IR dosage forms. Can you clarify for an oral solution and lipid, as those do not need BE studies?
For the solutions or lipids, yeah, biowaiver approach may be feasible and eligible as per the CFR. So if the generics formulation contain the same active ingredients and the differences in the excipient will not impact the bioavailability performance, in vivo performance of the products, in vivo B studies could be waived. However, based on the totality of the information, the data, if there's any concern regarding the bioavailability, in vivo B studies would be still needed depends on the submission. Thank you.
All right, thank you so much, Dr. Zhang. We'll move on to a question for Heather now. Will the FDA stop reviewing suitability petitions once the goal date has been met for the fiscal year?
No, we will continue to review suitability petition requests as resources allow.
All right, thank you so much for that response. And another question for you, Dr. Boyce. Why does an applicant need to conduct both a fasting study with water and without water for a proposed or for an ODT proposed product?
So this is dependent upon the intent that the ANDA applicant is looking for their labeling. If they are intending to have their ODT label, their product label indicate that the ODT product can be administered with or without water, additional bioequivalence study and fasting conditions may be necessary to test both conditions with and without water for the ODT product, because these are highly specialized dosage forms where water can impact the bioavailability of the ODT product. So for that first applicant trying to do the tablet to ODT switch, they may it may be recommended for them to do both of those studies.
All right, thank you so much for that response. Now we'll do a question for Karthika. How do I know if the design of my device constituent part is acceptable for submission in an ANDA referencing a particular RLD that is a DDCP?
Thank you for that question. As noted in the CA guidance, or the comparative analysis and related comparative use human factor studies guidance, the generic device user interface can have certain differences from the RLD, but they do need to be identified, characterized, and justified. However, if you still have questions regarding specific device design features of the device constituent part, we do recommend that you submit a controlled correspondence, or if your product is a complex product, you can include the question as part of a product development meeting. We do recommend that you do this during the early stages of your product development to obtain agency feedback. Thank you.
All right, thank you so much for that response. And then we have a question that is addressed to Dr. Jihong Shun. If a PSG has not been published for a drug product, can the clinical pharmacology studies conducted by the RLD program be referenced to consider for the selection of study subjects and dose?
My answer is yes. The clinical pharmacology study in healthy subject conducted for the RLD program can serve as key information or part of that for study population selection if available. I'd like to remind that it should still consider all available information and any additional component on the BE study setting too. You would also need to think about one factors. Healthy subject study for NDA program might be conducted under somewhat different setting from BE study, such as a confinement period as well as intensive monitoring methodology too. If you have any question on study population selection, you can still pose the question to agency using control correspondence and pre-ANDA meeting request. Thank you.
Can I add to that? So in addition, the clinical endpoint study, a clinical endpoint time point may be different for a BE study from what's in the RLD labeling. So just be mindful of that, like what is optimal there. You want to look at the PSG carefully to see what we recommend there.
Thank you so much for those responses. We'll do another question for you, Jihong. When a lower dose other than the highest strength is selected due to safety reasons, do you recommend the lowest strength as a PKBE study dose?
The lowest strength would be relatively safer than other strengths. However, one dose of the available strengths, including a middle dose, can still be chosen if the use of that strength in healthy subject is acceptable from safety perspectives. Likewise, some PSG recommend the middle strength as a study dose based on reported dose safety relationship assessment. In my presentation, I included 3 drugs which recommend a lower dose due to safety reason. It was aripiprazole tablet. This drug PSG recommend a middle dose, 10 milligram, as an optimal B study dose, whereas the other 2 drugs recommend the lowest strength due to safety reason. I also like to emphasize that if you have any uncertainty on the safety when using a higher dose rather than lowest dose, you should still choose the lowest dose. Thank you.
Thank you so much. Now we have a question that looks like it could be addressed for MJ, who was part of our panel. I would like to understand whether FDA also considers PSGs published by EMA for the same products. What different approach was EMA follows compared to FDA?
Thank you. So when it comes to so if I may kind of step back and address some of the major differences between the EMA versus FDA at the current situation, and we may see less of differences once the ICH M13A gets implemented. As you know, for the IR, immediate release products, oftentimes FDA may recommend both fasting and FAT-BE studies, whereas EMA may recommend just one study. Regarding the modified release products, and there was a very good meeting last week, a GBHI meeting, as well as a QRI meeting on the MR products last week. And I believe those slide decks may be available for people who may have attended. So kind of going back to your question about the EMA versus FDA, and when it comes to PSG development, from our end, when we develop PSGs or try to revise PSGs, we do kind of go back and see what the other agencies may recommend. And we try to understand if there may be some differences in terms of scientific or clinical perspective, and if there may be some significant differences, such as, like, for example, we had recent example of a study population for certain products. We realized that the EMA may recommend healthy or patients versus FDA may recommend the other. So we have been having some communications with our EMA colleagues to understand their rationale, and we also try to share our assessment and where we are coming from. But we try to utilize the all available information and try to kind of come to an harmonization, and we understand that coming to an harmonization will be very much beneficial for the generic applicants. So we are very mindful about this. So more to come when the ICH M13A is implemented, and I believe applicants will be very happy and excited that there is a harmonization, at least for the IR products, and we hope to continue with that effort for the modified release products as we move forward. Thank you.
Thank you so much for that response. Now we'll address a question that is in for Dr. Leah Filati. What if the USP monograph for our proposed generic drug product has a dissolution method that uses water? Can we use that dissolution method for our proposed generic product?
Hi, thank you for that question. These methods in the database are recommendations, even though there is a USP method with water. We would like to see the method not using water due to the lacking of the buffering capacity of water. So we would recommend that you try different media just to see how your product behaves. Thank you.
Great, thank you so much. And it looks like we might have time for one more question. This will be addressed to you as well, Leah. What if our proposed generic drug product does not have the same dissolution profile as the reference product?
Yes, if your proposed drug product does not have the same profile as the reference product, for quality control purposes, that's okay. We're not really looking at the comparator to the reference. For quality control purposes, we just want to look at the profile for your proposed generic drug product and make sure that quality is maintained throughout the shelf life. Thank you.
All right, thank you so much. Looks like that's all the questions that we have time for. Thank you again to our presenters for all the excellent information. Now we'll have some closing remarks from Dr. Robert Landberger, the Director of the Office of Research and Standards within the Office of Generic Drugs.
Thank you, Nora. Very fantastic to see everyone here today learning about the PSG program. I think this is a fantastic part of the generic program. Makes a huge impact on the industry. It's really, I think, a highlight of transparency and clarity about how the regulatory system works. And hopefully during today's workshop, we got a really deep sense about the value that the PSG program creates, like, to understand why we write certain things and what that means, what you can learn about the drug development program. Also, what are the opportunities to take a different approach, right? Again, the PSGs they provide a clear path that FDA believes will be successful in getting a product to market, but it's certainly not the only way that you can bring a product to market. And FDA is certainly open to alternative approaches and evaluating them on a scientifically sound basis. And, we understand that sometimes, the PSGs, when we post a PSG, we post it because we're absolutely sure that that recommended path will lead to success. It doesn't mean that we think other paths are wrong. It's just that that's the way that we have that's the clearest and most reliable path for you to take. And so, we're very open to other approaches, but it might say, well, if someone were to do this and it were to work, then we would accept it. But we don't want to write a guidance that directs you down a path that we aren't confident about success. But if there are opportunities where you see that, that you're willing to take that uncertainty on developing an alternative to the PSG, we have opportunities through pre-ANDA meetings for complex products and control correspondences for other products, and our MIE meetings for modeling and simulation approaches to get input into alternative approaches before you submit them. The other aspect of the PSG program that I think just always surprises me is that there's in the industry sometimes a lot of nervousness about what happens when a PSG changes. And I think from FDA's perspective, we don't think that should be the case, and we are really trying everything we can to minimize any concern about what happens when we revise the PSGs. We've heard this many times from representatives of the generic industry. The PSG changed and now I'm really nervous, or something might go wrong with our development program, or maybe we shouldn't submit. We try to allay this in many ways, one of which is through the forecast list, right? And so in the forecast list, we indicate if a PSG revision that's upcoming is a minor revision. And minor revisions generally mean that if you followed the previous guidance, that would still be acceptable. The minor might add an additional option. It might be, improve the clarity or efficiency or consistency, but it's not intended to say the previous approach was incorrect or is going to lead to a rejection of your application. That's what minor means. So if it's a minor revision, you should be less worried about it. Major revisions are cases where we do anticipate that it could have a very significant effect, and we may ask for new data if you followed the old study. And major revisions can come from new understandings of scientific risk, or they can come from changes in the reference product labeling or characteristics of the reference products that are essential for you to match according to our regulatory framework. So sometimes there are reasons where major revisions are needed, but by putting on the forecast list major and minor, it's really strongly intended to not make you nervous when you see, so I'm working on a product and there's a minor revision coming. That should not be a significant concern. We also have a program, if you are involved in an in vivo program, a more extensive program, and the PSG changes, even a minor change to an in vivo program, you can use the PSG teleconference to get a 30-day conversation with FDA. Really, if it's a minor revision, we're just going to reassure you that it's going to, that it's probably going to be okay. But if it's a major revision, to really talk through and get some input on the alternative approaches, and perhaps if there's additional data needed, or maybe in your situation there's not. But really, to allay that uncertainty is if you see a PSG come out and your in vivo development program is underway, you can use the PSG teleconference program to get clear, immediate understanding of the situation. And again, it's our intention that we revise PSGs to add new, more efficient approaches to keep them up to date so that you're not surprised. We don't want revisions to be a barrier to efficient pharmaceutical development of generic products. And so, we're also willing to hear other suggestions about ways that through transparency and communication, we can minimize the sort of idea that PSG revisions negatively impact generic drug development programs. We want the PSG to be up to date and current, and really hope that this webinar has helped you understand that even more clearly. And so appreciate all of the staff from ORS and OGD and OPQ that participated in this, and our SBA colleagues for organizing this webinar. This is a fantastic resource for helping you understand our PSG program. So thank you all for your interest and engagement with the generic drug program.